tonight we have with us Skyler Hedden from the Arizona Game and Fish Department. And I met Sky several months ago when he came to a meeting for the Virgin River Coalition. And he did an excellent talk there. And I thought, boy, this guy would be good to have at our speaker series. So I asked and he said, sure. So we are again lucky to have Sky here with us tonight.
All right. Yeah. Hello, everyone. Like Brenda said, I'm Sky Hedden. I'm a Colorado River Basin coordinator with Arizona Game and Fish. So basically, I'm the one of the fish biologists with the department, and my job is basically managing with others all streams within the Colorado River Basin. And so one of those tributaries is the Virgin River. So that's the talk I'm going to be giving today. So here on the title slide here, I just have basically all of the native fish that occur in the Virgin River watershed. So on the top left there, we have the woundfin. On the right, the Virgin River spine dace. Below that, the speckled dace. So these are three fish that we classify as small-bodied fish. So they're, as adults, they're generally less than 100 millimeters, so no more than about 4 inches long. And then the other three natives, they can get quite large: the Virgin River chub, the flannelmouth sucker, and the desert sucker. As adults, they can get upwards of about 16 inches, so about 1.5 feet. And kind of just give a brief outline of my talk today. So there's really 3 parts to the talk. So first, I'm just going to kind of give some background on the Virgin River and the fish that occur there and some challenges that they face. Next, it's going to focus in on the management that we've been doing in the system, both in the upper Virgin River in Utah and in the lower Virgin River in Nevada and Arizona. And then lastly, kind of just focus in on the lower reaches of the river in Arizona and Nevada to really give our management goals or future direction of how we're going to manage this native fish community. So kind of just give a background to the Virgin River. Here's the Virgin River watershed in this light green. So the Virgin River was once a major tributary to the Colorado River, but it now drains into Lake Mead. And we can see the main stem flows across portions of Utah, Arizona, and Nevada. And so looking at the headwaters and Zion National Park, this is kind of your picturesque mountain stream. It's bouldery, clearer water, cooler water. As you move down into Utah and Arizona, you enter the Virgin River Gorge. This is canyon-bound reach. The river's typically sand in this reach and cobble mixed in. And then when you get down near Mesquite and downstream into Nevada, this is where the floodplain really opens up, and it's a much wider river, shallower river, and a sandy river down in this reach. And so, like I said, the Virgin River fish community, it's a very unique fish community, and that's for a couple different reasons. So first, we have species such as the speckled dace. So this map shows its native distribution in this orangish color. And so this species is very broadly distributed across this western United States, where you can find populations from Canada all the way down to Mexico. And so it's unique in the fact that even though it's broadly distributed and you can find it in all sorts of streams, it's a unique fish because it's a fish after a disturbance, so a drought or a flood. It's a fish that can move very long distances and recolonize areas before any other fish gets there. So it's just a real small fish, but it's capable of these long dispersals. And that's why it has such a broad distribution, because they're great at moving around. What makes the fish community also unique is you have species such as the woundfin. And so if we look at this map, we see the orange coloration is much smaller. These little dots in Nevada, Utah, and Arizona. And that's because it's an endemic species. So it evolved in these systems and it really never moved out. So the Virgin River is actually the only place in the world where you can catch this fish. And that's the same for Virgin River spine dace. So it's a species that is endemic to the Virgin River. Nowhere else in the world can you find these 2 species. And in fact, it's the same for the Virgin River chub. So like I said, there's 6 native species. Half of that, 3 of them, you can only find in the Virgin River system, which makes it really a unique system. And because these species only occur in this part of the world, they're very threatened. If we lose these populations, they're extinct forever. And so because of that, they're protected generally. So 2 of those species, the woundfin and the Virgin River chub, are listed under the Endangered Species Act as endangered, and the other species that's endemic to the Virgin River, the spine dace, it was recently proposed for listing. And so then the other three species, they have a broader distribution across the western states. But for the last 20, 30 years, we've still been seeing large population declines of these species, but not warranted to be listed quite yet. And so what we can do is the best way to learn about these species and to manage these species is to simply sample them. So if we go out and collect these fish species through time, we can understand how they might respond to droughts and to floods and to human disturbances, such as the introduction of other species. And so when we sample the Virgin River, we're typically doing what's called seining. So it's a long net on two poles, you drag it through the water and you simply scoop up the fish. And so this is the most effective way to sample fish in these larger sandbed rivers that are typically turbid. So the water's cloudy or muddy at times. The fish can't see it coming and you can easily scoop them up. And so through decades of research within the Virgin River, there's been identified a lot of negative interactions or negative pressures being put on the native fish community. And so these challenges for the native fish are primarily the introduction of non-native species. So like I said, the Virgin River was once a tributary to the Colorado River, but that was dammed with Hoover Dam and it now flows into Lake Mead. Well, Lake Mead is managed as a sport fishery. So a lot of non-native fish from the Midwest and the eastern parts of the states are stocked in there to give anglers sporting activities. And because of that, since the system is connected, a lot of these fish can just swim up the Virgin River. And so a lot of these introductions have been from Lake Mead, fish swimming up the river into portions of Arizona and Utah. Another major challenge for native fish in the system is altered flows. So here's just a picture of showing the same river reach in the Virgin River Gorge with water and without water. And so what's driving altered flows? It's a lot of factors combining, such as climate change, water diversions, and human consumption of the river. And so fish typically need a certain flow. We call it a flow regime, a pattern of flow that fluctuates through time over the year. These fish have evolved and are used to these changes in flow during spring, where there's typically higher flows, summer, where there's typically lower flows, and monsoon storms, where there's variable flows. So the species have evolved to this, and they actually prepared, and then they respond positively when we see these natural flow regimes. So these natural floods that roll through, and then that oftentimes cue their spawning. Well, we've disrupted that a lot by building reservoirs, by extracting water from the system. And then the third major component is the altered habitat. So not only have we reduced flows, but we've actually changed, because of reduced flows, the habitat has changed, and we've also created structures that physically change the habitat. So example here is just a low water dam, where basically we change the river habitat. We're slowing the water down. We're creating shallower reaches in a lot of the river. So these are really the three major challenges that the Virgin River native fish are facing. And kind of just dive a little bit more in depth so you understand this. So in terms of non-native fish, like I said, these are sport fish that were put into Lake Mead and then swim upstream. And so the picture on the left is a seine hull. And what you see, it's full of channel catfish, common carp. So these are all non-native species that were never in the Virgin River until humans moved them there. And then the number one threat is what we think is these red shiners. So it's just actually a really beautiful small minnow species native to the Midwest, has these bright color red tails and kind of a bluish back, these spawning males. But the problem is these non-native fish, they compete with our natives. They eat the same resources, they take up the same space, and then they also predate on them. So even though these small minnows, the red shiner, they can actually eat the native fish eggs and eat juvenile fish right after they spawn. And the reason we think these are such a problem, because we have long-term data. So like I said, the best way we can understand on how native fish respond to different things is by going out and collecting data. And so we've actually have this multi-decade data set from the Virgin River that dates all the way back to the mid-70s. And so this graph is just showing wound fin densities and number of fish captured through time. And so these gray bars are just the catch rate. So how many fish you catch in each one of those seine hulls we pull up. And that black line is just the raw number of wound fin we catch in the river. And so what you see is throughout the 70s and early 80s, it's pretty consistent where you're catching 100 to 300 fish per sampling event. But then we see in the mid to late 80s with this drop off, where essentially the fish is voided from the river. So this site is actually in Littlefield, Arizona, near the Beaver Dam Wash confluence. And it just so happens it coincides right when a lot of these non-natives invaded the river. So that arrow indicates right when red shiners actually invaded the river from Lake Mead. These red shiners are stocked as a bait fish. And then typically when people are done with their bait bucket, instead of disposing the fish, they just dump them in the lake. And so the population grows and then it begins to disperse upstreams. And so basically when this non-native fish invaded the river, one of our native fish essentially disappeared. And so this is a large reason why that fish is currently listed as endangered, because it's really difficult to find these species in the wild anymore. But it's not just woundfin that we see this negative interaction. So we can plot red shiner density. So how many fish per 100 meter of stream from these large data sets. So each black dot would represent a month and a year that we went out and sampled the river. And then we can just plot that on the vertical axis. We'll compare how many red shiners did we catch here and how many native fish did we catch here. And we see this negative line. So the more red shiners we catch in the river, this non-native fish, we get less and less native fish. But in some years where there's a lot of native fish present, those are the years when we actually catch the fewest amount of red shiners. So this kind of describes this negative relationship that these non-native fish are having negative pressures and decreasing native fish densities in the river. And so just one more graphic to kind of just show you this. So these are 4 sites in the lower Virgin River, Cedar Pockets being within the gorge, Beaver Dam Wash by Littlefield, Mesquite Diversion just upstream of Mesquite, the city of Mesquite, then the Riverside Bridge just downstream of Mesquite. And so we can plot these black bars. So these are proportional fish captured. So black being non-native and gray being native. So if these bars were half black and half gray, that would indicate half the fish we go out and when we go out and sample the river are native and half are black. What we see is in the lower Virgin River in this year, they're just all black, which means that when we go out, generally almost all of the fish that we captured are these non-native fish running up from Lake Mead, establishing populations and reproducing the Virgin River. So basically we see a river completely changed from what historically was there. We have this native, very unique fish assemblage. Now we just have this very common non-native fish that you can find in basically all around the world. And so another challenge is one way we can fight these challenges is constructing these non-native fish barriers. And so here's some pictures of those that we actually have distributed throughout the Virgin River. So what these do is basically they block all fish movement. So any fish downstream can no longer access those upstream habitats. But when we do this, we divide the river up into more manageable reaches. And so we look at a map where we have, so these gray squares represent either diversion structures that act as fish barriers or management strategies where we built fish barriers to try to prevent further spread of fish upstream. But like I said, is we divide the river into these manageable chunks where we can focus upstream and systematically work our way downstream and try to eliminate these non-native, to basically renovate the streams into these native fish communities. And the way we remove these non-native fish is through the application of what's called rotenone. So it's a chemical that you can add to the river. So the picture on the left, this is a big 50-gallon drum. You add this rotenone chemical to it. It's this milky cover when it comes out of the water. And basically, you drip it into the flowing water. It mixes with the water and drifts downstream. And what it does is basically it interacts with any organism that has gills. It basically interferes with their cellular respiration. So if you had gills and you're in the water when this is dumped in there, you can no longer breathe. So basically, they suffocate to death. And so what we do is, so the picture on the right now is those 2 black squares are going to be 2 of those barriers. So we're going to focus in on this reach where we know fish can no longer move up. Upstream is safe. There's no non-natives up there. And we have these red dots, these barrels located throughout the stream. And we drip this chemical and we basically eliminate all organisms that have gills in the river. And so then we eradicate these fish, and then now we can move downstream into the next reach and repeat this. And so when you apply this chemical called rotenone, basically this is what the stream looks like afterwards. You see all these silver fish, those are all red shiners that were dead. And so what makes this a useful tool is because it can completely wipe out a population. And since the river, a lot of times in the Virgin Rivers cases, it's just full of non-natives, we're not worried about killing any native fish because really there aren't barely any left in the river in these reaches. And then what's great about this chemical is we can at the downstream end, we can apply a different compound called potassium permanganate. It has a purple dye in it, so you can see it when you put it in the water. And so what it does is it binds with that rotenone and basically purifies the water. So then that way you're not having this long reach of dead fish. You can basically add this at the bottom, and basically it gets all the rotenone out of the water, and then all fish downstream all the way down to Lake Mead will not die. And so they've done a lot of these treatments up in the upper Virgin River, like I'll discuss it a little later in my talk. But for decades now, they've had the successful eradication of non-natives. And so we can plot these same bar graphs, again, proportional data, each year of sampling. Gray is native, black is non-native. Again, if it's half black and half gray, that indicates half the fish you catch are native, half are non-native. What we see up in Utah after these rotenone treatments, for many decades now, we only are catching native fish. We do have some small black bars there, but these are really benign non-natives that we're not too worried about, that are escaping from golf course ponds, that they get out, but they really cannot reproduce in the river. So they're not really a large concern like their red shiners. So it's a really effective tool, and it can have great results when done properly. And then in terms of altered flows, like a challenge for native fish is, like I said, is not only are they evolved to respond to different flows and reproduce and live in these different flows, within this portion of the United States, it gets really hot. And so when we extract water, not only do the fish not have water in some reaches, like this reach here, we could see it completely dry, but in reaches that hold water year round, the water gets so hot that it exceeds the temperature, the maximum temperature these fish can live. And so essentially they perish. And then also, like I said, these altered flows or altered flows lead to altered habitat. And so a great example of this is we can just look at this a diversion structure near Mesquite. And so these diversion structures, they provide great human benefit. So we extract water typically to grow crops, and they have water rights to do so, legal rights to extract the water. But oftentimes, it's not the most efficient system, and we can see that from a figure like this. So on the right image here, we have the Virgin River. We can see it's a wider river, plentiful water, and then we see that black line kind of right in the middle. That's the diversion structure, the canal there labeled kind of going off to the bottom of it, extracting water. And what we see is below the diversion structure, the channel's much smaller and much narrower. There's less water because they're diverting water. So the problem is not the fact that they're diverting water. The problem is that sometimes these systems are not super efficient, and oftentimes they're diverting water when the crops need it the most during the hot summer months, which also are when native fish need it. And we can actually plot these habitat depths. So that's the aerial image, but we have data to back this up. So 2 sites above these diversion dam structures, we can plot the stream depths across a 1 kilometer reach. And so these are 2 different sites here where basically they're called depth profiles. So different colors indicate different depths. The redder the color, the deeper the water. So in reaches where we don't have diversion structures, we see there's a lot of this mix of habitat. There's deep water, there's shallow water, and it's dispersed throughout the whole reach. But if we look at 2 sites below these diversion structures, what we see is it's basically all shallow habitat. We're only seeing these blue colors, and we're missing a lot of these red, darker colors. These are the deeper waters where fish can run and hide to during those hot summer months and kind of protect themselves from that heat. And then also it protects themselves from bird predators trying to eat them as well. So that's kind of the first overview of the talk, just introducing the fish and some of the challenges they face. And now I'm going to move into part 2 of my talk today, and that's kind of the bulk of it, the meat of the discussion, and the data is going to be in this part where we're comparing the upper Virgin River up in Utah to the lower Virgin River down in Nevada and Arizona. And so it kind of just a picture showing you the transition as you move from upstream on the left down to downstream. So we have clear habitat differences, but not only we have habitat differences. Oh, before I say that, I just want to just point out this slide. So all of the information I'm showing today, especially the data presented, it's been a multi-collaborative effort between multiple state agencies since the river flows across three state boundaries, federal partners, nonprofits, and then also some consulting firms that provided either funding or data for this project. And so for this portion of the talk, like I said, the river is divided in the upper Virgin River and the lower Virgin River. And so that's typically upper is at Utah, lower is Arizona and Nevada, just to make things clear. And in the upper Virgin River, they've done extensive native fish management. So like I said, they have several constructed fish barriers to prevent more non-natives to move upstream. They've done numerous eradications, so applying this rotenone to the stream to kill off these non-native fish. They have native fish stocking programs, so they extract native fish, hold them in a hatchery, grow them, and then re-put the juveniles back in the system. And then they have these elaborate pump back systems, so where they can actually pump water from reservoirs, that's cooler water, that's deeper, that's not getting hit by sunlight, pump it into the river and cool temperatures down. So during these hot summer months, with climate change, we see these stream temperatures have been rising and rising. They can actually get cool water, put it in the river and cool it down to make sure that temperatures remain suitable for our native fish. Now, so this is very different than what's been occurring in the lower Virgin River, which really it's been in this planning and researching stage for the last decade or so. So a very less management activities compared to the lower Virgin River. And so for the purpose of this part of the presentation, there's really two main questions that I'm going to dive into. So the first one is, what are differences we see in the native fish communities in the upper and lower Virgin River based on these very stark differences in management over the last decade or two? And is the lower Virgin River even capable of sustaining similar native fish densities as I'm about to show you that they see in the upper Virgin River? And so for this portion of the talk, there's going to be 12 sites that I'm going to have data from. So 6 in the lower Virgin and 6 in the upper Virgin. And so then the upper Virgin, all of this is collected by the Utah Division of Wildlife Resources. So basically what they do is go to a site and then they sample it every single fall for several years. And we basically have the identical data set in the lower Virgin River collected by a different entity. That's again, so it's these very comparable data sets from the same time period of the same years through time. So what we can do is we compare these data sets for similarities and differences. And the best way to do that is this tool called non-metric multidimensional scaling. So it's a fancy statistical term for basically what it means is we're trying to visualize levels of similarities in data sets. And so I have an example just to make sure we're all on the same page, because if you see these graphs, they can be overwhelming. And a great example is to show, basically you can plot data on anything within these graphs. So for instance, we can plot the US politicians' voting history. So basically what this shows is each dot would be a politician. And where you fall out on this graph is the voting history of all of the things you voted on in the past. And so it's a great tool just to visually look at very complex data. So a lot of different people voting on a lot of different things and just put it on one graph. And so what you see is basically Elizabeth Warren is on the far left of the graph. Donald Trump is on the far right. These two individuals never agree on anything, so they're very far away from each other. That's basically what that's showing. The closer you are, the more likely you agree. And so if two politicians voted the exact same way their whole careers, those dots would fall right on top of each other. They'd have identical similarity, they'd fall on top. The further away, the less similar or the more different you are. For example, like Joe Biden and Mitt Romney, still very different individuals, but they're a lot more similar than Donald Trump and Elizabeth Warren. So they're closer together. They're in the middle of that graph. And which makes this a powerful tool is we can plot all sorts of different things on these graphs. We can plot these circles to show you that generally all of the Democrats fall in here and all the Republicans fall in there. So then you can single out which Democrat, which Republican actually falls outside of how their party generally votes. Another great thing is you can put on what's called these point scores. You can basically look at all the bills they voted for that were either pro-choice or pro-gun or pro-freedom and see where do you fall out. So generally what you see is the Democrats fall out on the pro-choice side, and generally what you see is the Republicans fall out on the pro-gun side. But one thing they agree on, it lies right there in the middle of the graph, is they all agree on pro-freedom. So those points that lie in the middle, they basically say everyone agrees on that. And so basically this is what we're going to do for this fish community data set that we have. So like I said, we have almost a 10-year data set where we're sampling a lot of different streams and a lot of different years. So it's the same premise. Is we might go out to the site, Mesquite diversion upstream of Mesquite. And so each dot would represent a year we sampled that fish. So here's 2012. And basically where you fall out on this map is the fish that were collected at that site. So for instance, we caught 500 some red shiners, 120 some flannelmouth sucker, and so on. And so based on what fish you catch is where you fall out on the plot. And so another dot might be the Utah reach from a different year. And again, so like I said, they've eradicated a lot of the non-native fish upstream, so you're going to get 0 red shiners there and so on down the list. So these dots are kind of farther away from each other because they have different fish that occur at these sites. And it's the same premise. You can plot these circles to basically show that generally the Mesquite diversion falls within this area. No matter what year you go out there, you're generally going to get this fish community. Same with the Utah UDWR reach number 6. And then we can clump it even further and comparing the lower and the upper Virgin River sites, so basically Utah sites and Nevada and Arizona sites. And then we can plot these point scores, species scores, to see, all right, generally these species are found at these sites and vice versa. And species in the middle are found at all sites. So if we actually plot this data on this graph, what we see is the first thing we see is clear differences in upper and lower Virgin River fish communities. And so this p-value, basically the smaller it is, the more likely it's different. And so we see this very small p-value, and clearly these circles do not overlap. All the dots on the right are the upper Virgin, all the dots on the left are the lower Virgin. And when we plot these species scores, so green species are our native species, and red species are these non-native species. What we see is generally all the red species are on the left side of the screen, and none are on the right. That's because Utah has eradicated a lot of these non-native fish through the application of this rotenone. So because of that, we get a lot more non-natives down in Nevada and Arizona. But the interesting thing you see is there's a lot of natives that fall right in the middle of the graph. Virgin River chub, speckled dace, desert sucker, flannelmouth sucker. So these are species that you actually catch at all sites. And in the upper Virgin River, it's just 2 species that you generally don't catch down in the lower river, the spine dace and the woundfin. But like I said, you can take this one step further and look at individual sites. So like I said, we have 12 sites. So if we break them apart of all sites, so it's probably hard to see on your computer screen, but in the upper left there, it's going to be a map of all of the sites. L1 through L6 are in the lower Virgin, and then U1 through U6 are in the upper Virgin River. And so basically, first what we see is on the right side of the screen, all those circles are falling right on top of each other, which means it doesn't matter where you go in the upper Virgin River, you're going to catch the same fish community every time. But what we see in the lower Virgin River, it's a little more interesting. Those circles don't overlap, and they're almost structured L1, 2, 3, 4, 5, 6, just as they are down to upstream. So what this shows you is those L6, L5, L4 sites, those are more similar to the Utah sites. And as you move downstream towards Lake Powell, we get less and less native fish and more non-native fish. And so we could take this, but these results aren't shocking. Upstream, they got rid of non-natives. Downstream, we haven't. Downstream, we have the source in Lake Mead that they're moving out of Lake Mead. So of course, these lower sites going to have more non-natives. So what if we just remove those from the data set? We ignore non-native fish, and we're only interested in now looking at the native fish community to see if there's any differences now. What we see is when we run the analysis with just those 6 native species, we see there's a lot more overlap. Those circles between upper and lower Virgin River become much closer together. So our p-value, which is a statistical test to tell us, are they different? It still says they're different, but they're a lot more similar. We can see that on this figure. Again, we can plot those point scores, And what we see is a lot of our species, again, just like in that other graph, those native fish that we catch at all sites, speckled dace, flannel sucker, Virgin River chub, desert sucker, they fall right in the middle between upper and lower. And what's really driving these differences are these two species, the spine dace and the woundfin, that you catch upstream, that you don't catch downstream. Not surprising, these are those two small-bodied individuals that probably compete the most with red shiners. So in reaches where you don't have these fish, they are doing better than reaches where you have these non-native red shiners. Again, we take that one step further and plot each site. And so what we see in Utah, again on the bottom there, it's hard to see, but all of those Utah sites are right on top of each other. Basically, it doesn't matter. The native fish community is the same at all those sites. But in the lower Virgin River, well, again, we see this very clear discrepancy that the L1, L2 sites, those furthest downstream sites, are very different than the rest of our sites. And so those furthest downstream sites actually have less native fish than our sites as you move up the river. And so another way to look at this is just to plot how many native fish do we catch per 100 meters of stream. So in Utah, it's like I said, it's consistent. You get anywhere from about 3 to 10 native fish per 100 meters of stream. We continue this downward trend, we still see these values remaining high, even in reaches where we have non-native fish, but then we see this sharp decline as we go downstream. And so to answer that first question is can we sustain high levels of native fish in these lower reaches, even though the habitat's so different? And the answer is yes. But in these very low reaches, we see this drop off. And if we try to figure out, well, what's causing that severe drop off? Because we know there's non-natives throughout the lower Virgin River, but what else is different? And so we can look at, again, these depth profiles. So here's 7 sites located along the lower Virgin River, looking at these depth profiles. And then in parentheses, those sites correspond to the fish graph on the right. And so just looking at those depth profiles, what you see is on those sites on the left, we see all those blue and green colors, real shallow water. And those are the sites that coincide where we have our lowest native fish present. So not only do we have a bunch of non-natives present, but we have very shallow and simple habitat, which the natives don't prefer. And so that kind of leads me into the last portion of my talk today is, so now what? So we understand we understand a lot about the native fish community, what they like, what they don't like. So what are we going to do about it now in the lower Virgin River, where we have all of these problems and historically not a ton of management relative to the upper Virgin River? And so basically through the works of states and federal partners and the Virgin River Coalition, nonprofits, we all came together with this common goal of trying to improve the native fish community in the lower Virgin River. So we have this two-phase of plan approach that we hope to implement over the coming years and decades with both short-term goals and future actions. And so I'll kind of walk you through each of these short-term goals and future actions here. So first and foremost, we want to work with the irrigators to see if there's any improvements we can make to their canals without reducing the water they extract, but make it maybe more efficient or more productive, or at times of the year where we can benefit both the irrigator and the native fish. So because like I said, we can look at these depth profiles, we can look at imagery, it's clear that these canals are having an impact on the native fish community. We also are going to explore high flow pulse releases. So like I said, we have a lot of data from the Virgin River, so we know a lot about the native fish, but we also know a lot about the non-native fish. So this non-native red shiner, one thing we learned is they don't like high flows. They simply can't withstand them, either whether they get the young get blasted downstream or they just have low survival when these high flow events occur. Basically what we see is this negative relationship. So this graph here is just showing you the peak monsoon flood in a given year, and then the percent change in red shiner density. So typically a monsoon flood in the Mesquite area produces a discharge around 600 cubic feet per second. So typically the river is running in the summer months around 50 to 100 cubic feet per second. So these are these moderate floods. So basically what we see is the red shiners at these densities, they're neutral. But if we have these years where we don't see these monsoon floods, we actually see this percent increase in the amount of red shiners in the river. But when we do see these floods, so for instance, If we increase the CFS by 300, if we increase the discharge, the amount of water flowing through the river by 300 cubic feet per second, we see this 50% reduction in red shiners. So they do not prefer these high flow events. So we can somehow manage the river to create these high flow pulses and basically wash these non-natives to reduce their numbers. And then lastly, in the short-term goals, we're hoping to protect and enhance any habitats within the lower Virgin River to basically help the native fish and hurt the non-native fish. And then for future actions, these are probably multiple years, decades down the line. We're exploring the idea of creating another fish barrier down closer towards Lake Mead to prevent further fish from invading. And then once this is done, hopefully apply rotenone to eradicate non-native fish through these lower portions of the river where we see really high abundances of non-native fish. And then lastly, can we kind of restore the habitat? Can we remove any barriers that are altering the habitat? That way we can allow native fish to move around and get more of this flowing river through these habitats. And so kind of just in conclusion, the key takeaways of this talk today is first, the Virgin River contains a very unique fish community. We have half the fish you can find nowhere else in the whole world. They face several challenges though to persist. But luckily in the upper Virgin River, they've been very successful and we know management techniques we can do to promote or to enhance the native fish community. And then down in the lower Virgin River, we've done a lot of research and we're in planning, we're hoping to mimic those upstream successes. And then lastly, really what's driving a lot of this success is these collaborative partnerships where we have multiple agencies and concerned people coming together with common goals. And really that shows you that past and current management activities should ensure that this native fish community remains intact. And so that's all I have for today. So if anyone has any questions, I'll gladly take them.
Okay, Sky, I do have two questions in the chat. If you can ask, or if you can answer them or possibly point them in the right direction. The first being, are there any planning efforts being made for the effects of the St. George population boom on Virgin River hydrology and native fish species?
Yeah, so that's a good question. So the Utah Division of Wildlife Resources, they have a headquarters there in Washington. So just right next to St. George, it's the Washington County. They have been working with the city of St. George, I can't remember what it's called, the Water Conservancy District there. Basically, they've been working with the city for decades now, knowing that this is going to be a problem, that the river is going to face more demand from humans. So the city and the managers up in Utah have been preparing for this. And so they've actually done such a great job of managing the native fish community that probably it's basically allowed these species to still exist. And we're benefiting that from downstream. And so, yes, they're planning very closely with the city and water users to ensure that we can have both. We can have a population, a healthy and growing human population while ensuring there's enough water and these fish are protected. So like I said, 2 of the species are actually listed under the Endangered Species Act. So they also have legal rights that anyone will face if they try to extract more water from the system.
Okay, here's one. Since amphibians don't have gills, they would not be affected by the rotenone,
yeah. So yeah, that's correct. If the species doesn't have gills, they're perfectly fine. So yeah, an adult amphibian, minimal to no effects at all on those species.
So does each species have their own range, i.e. how far up into the headwaters does each range?
So every species, basically what dictates where they're at in their native reaches is primarily temperature. So species such as the speckled dace, that species, it can withstand very cold temperatures and very hot temperatures. That's why you have such a large, broad distribution where you can find populations all the way up in Canada and all the way down in Mexico. And so the same thing, so speckled dace you'll find all the way up in Zion and all the way down into Lake Mead. Other species, they kind of have this thermal range that they target in on. And so like the woundfin, Virgin River chub. So that you'll find those basically basically from a little upstream of St. George all the way down into Lake Mead. Flannelmouth sucker, again, it's a species that's broadly has broad distribution, so it can move around and tolerate cold and warm. So you'll catch them all the way upstream as well. So it depends on the species, yeah, and that's primary temperature is what's driving that.
Do algae blooms affect oxygen levels in the Virgin River?
To simply put, algae blooms are not a huge problem in the Virgin River, so it's not a huge concern for Virgin River fishes.
Here we go. There is currently a bloom in Baker Reservoir. Is it spreading into the Santa Clara?
Yeah, so typically the problem with algae blooms are in standing water, where the algae can grow fast and grow very quickly and become abundant. Typically in flowing water, it's not a concern as much. The algae can't attach to things, so it basically just gets washed downstream. And then also, you'll have more turbulence in rivers, so like riffles, where you can actually more oxygen has chances to enter the water. And so generally speaking, algae blooms are a concern for reservoirs and not so much for streams.
Why are there warnings about blooms in the Zion Narrows then?
Yeah, I couldn't tell you the exact answer. My guess would, because so algae can grow in streams. It's not impossible. But typically they don't result in low levels of oxygen in rivers. So that would probably be like a human safety hazard, where if you ingest the water and the algae is in there, you could get sick or your pets could get sick, but it's not going to be a problem for fish and oxygen levels. That's typically what kills fish, is the low oxygen. But they can hurt humans or other individuals if they digest them.
And someone put in cyanobacteria. Is that what
Yes.
killed?
Yeah, that's the bacteria that's typically the dangerous, or the one that you don't want to swim in or eat or have your pets drink water if that's in the water.
But Sky, I wanted to thank you so much for your presentation this evening and for your amazing amount of knowledge that you shared with us. And we hope to have you back again sometime. And you just did a great job tonight. And on behalf of everybody here at Friends of Gold Butte, I thank you.
Yeah, of course, anytime. Thank you for all listening.
You bet. All right. Good night, everybody, and take care. Everybody stay safe.